Hello, my name is Shurosri Pal. I am a Prime Minister's Research Fellow, and we are discussing this question from GATE Examination 2018, Paper XLS, question number 2. The question states, which of the following immune system components can function as an opsonin? A. Antibodies, B. T cell receptors, C. Histamines, and D. Interferons. For more information on opsonin and these immune system components, you can follow this NPTEL course, Immunology. Now, what is opsonin and what is opsonization? So we know that there are three functions of antibodies: neutralization, opsonization, and complement activation. So we know that antibodies can actually neutralize antigens. So what do they do? These antigen antibodies can form complexes. So this antigen antibody complex becomes a large complex, and which is why this antigen antibody complex can precipitate and the antigen can be neutralized. So it cannot remain in the system anymore. So that is how the antibodies neutralize the antigen. The second pathway of the uh, antibodies, the effector function of the antibodies is opsonization. So opsonization means that the antibodies coat the surface of the pathogen in such a way that they actually um, they actually enhance the process of phagocytosis. So after this neutralization, the antigen antibody immune complex can be ingested by the macrophages. This after this opsonization process, after the antibodies have coated the pathogen, then that antigen antibody complex and the antigen pathogen complex, antibody pathogen complex, that can be ingested by the macrophages. And so this actually initiates the process of phagocytosis. And the next one is complement activation. So we know that there are complement proteins. So the complement proteins and the antibodies. So the antibodies can activate the complement proteins, and therefore the complement proteins cause lysis of the cells and ingestion. So we know that the complement proteins that can form the membrane attack complex, or the MAC. So we know that there are proteins like C5b, C6, C7, C8, C, and C9. These five proteins together form the um, membrane attack complex, which makes a pore on the surface of the pathogen and then lyses the pathogen. So these are the three effector functions of antibodies, and one of them is opsonization. So what is opsonization? Opsonization is nothing but um, cross-linking of the um, Fc receptors. So basically, the antibodies, they coat the surface of any pathogen. So when the free immunoglobulin is there, it cannot cross-link the Fc receptors. But when these immunoglobulins can um, when these immunoglobulins attach to the surface of the pathogen, so then what happens, that causes the aggregation and then activation of the macrophage, leading to phagocytosis and destruction of the bacterium. Right? So that is how the opsonization process works. So in among these following immune system components, antibodies can act as opsonin. So opsonin is helping the phagocytosis to begin, helping the phagocytosis to initiate, right? The rest of them, like T cell receptors, histamine, interferons, they cannot function as an opsonin. So our correct answer is A, and that's it.